Welcome, welcome, welcome. We are live. It has been a hot minute. Hi everybody, how's it going? I got a fun stream for you today. I know it's been a little while since there's been any new stuff on this channel, but I think I got a good show for you today. So, as the title says, we're going to be talking about the Neuron project, and we're going to go over what we did the last time we talked about this and what the plan for is this time, really. But before we get going, I'm going to just take a couple of seconds, just let everybody join, get the show going. How's everybody doing? Everybody having a good day? Yeah, we got lots of people tuning in. It's gonna be a good day, I think. Alrighty, so let's talk neurons. I think I'm just gonna get right into it. I'll do the usual spiel first, which is basically like there is a donation bar. It will go to supplies for this project. If you want to kick in, greatly appreciated. Don't have to, but it's always appreciated. Alrighty, let's get going. Neurons, the topic of the day. So, what are neurons? Well, I mean, it's your brain, basically. And, well, the point of this project is to connect neurons to a computer, which sounds a little weird, but it's surprisingly easier than you'd think. Oh, thank you, Connor. Greatly appreciate it. Alrighty. So, what is the end goal of this project? I want to make some neurons pass butter, because, I mean, be the mad scientist of your dreams, obviously. But no, seriously, this is legitimately the plan of this project if we can get this far, but we will see. So, first things first. Ooh, thank you. Someone else, Horchov, kicked in. Thank you very much. Greatly appreciate it. So, this was the last attempt at the Neuron project. You also notice the lovely new thumbnails. For those that haven't noticed yet, the entire channel got a bit of a makeover, and every single thumbnail on every single video has been updated. So, they all look very shiny now. Super fun. So this was the last project. So what did we do last time? Well, first things first, we made these arrays. These are glass microscope slides that we coated in a double layer of silver and then gold, and I chopped the top off of a 15 mil Falcon tube and epoxied it using BioSafe epoxy onto the glass microscope slide so that it turns it into a little culture dish. The idea being each of these little pads connect to a little tiny electrode inside the culture dish, and we're able to theoretically listen to the signals that the neurons are putting out. And in theory, we could feed signals in, interact with them, that sort of thing, which would be lovely, but we did not get that far. But let's go over what ended up happening. So thing number one, this was the seeding of the arrays. And this wasn't the point where things started going wrong. Things definitely started going wrong much sooner than this, but this spot was where we made a lot of errors. So while we were seeding the arrays, we used human neurons, which we bought from an online supplier, which they still exist, although their price for the neurons has, I think, tripled or quadrupled, so we're not going to be buying from them again. Yeah, we used way too many neurons for the size culture vessel that we had. We probably should have used about 1/10 of what we ended up seeding, but I mean, you know, it was our first try, so we had to just go with something. But you know, live and you learn. This is one of the many problems. So first, you can see each of these tiny little dots here are each individual neurons. So yeah, way too many neurons. They didn't have enough space to actually extend their dendrites and axons and stuff, so they couldn't actually make connections. They were just too dense. But the other problem is these are some of the electrodes. Ooh, somebody else kicked in. Rafi, thank you very much. Greatly appreciate it. These are some of the electrodes that were inside of the culture vessel, and you can see these big cracks. This is where the metal that we had sputtered onto the glass had delaminated. So not only was it going to wreck the connection, it meant that like neurons got underneath, and it's not what you want out of a neuron array, obviously. So yeah, that's where that was. Again, here's a closer shot of the overseeded array. Again, all of these little dots are all individual neurons. Way too dense. Way too dense. Ooh, we had a couple more people kick in. Thank you, 𝐎𝐿𝐴H𝐿𝐼. Greatly appreciated. Yeah, too dense. And even if we had succeeded, even if we had seeded the right amount of neurons onto the array and the arrays hadn't broken, which was the other problem, of the 4 arrays that I brought to the lab where we did this, 3 of them broke. So, you know, not a great success rate by any stretch of the imagination whatsoever. But even if we had succeeded, our method for actually listening to the pulses from the neurons was atrocious. Like, we didn't really have a solid plan. I had an oscilloscope, which we, you know, used some copper tape and kind of probed the electrodes, but we didn't really get anything. We got like a voltage, but that's not really what you want. I also had this thing, which is a spiker box. Ooh, Ayrton kicked in. Thank you very much. Greatly appreciate it. This is called a spiker box from Backyard Brains. It's meant for... Basically what you can do is you can either take a cockroach or a worm and you put 2 little pins either into their antennae or just like through the length, through the length of the, like, one end of the worm, and then you'll hear like clicks as the neurons fire. That's sort of what this thing is meant for. That was as far as we'd really gotten. So yeah, not really ideal. Okay, so what didn't work last time? We had insufficient culture techniques, we had low quality arrays, bad electrical solutions, we tried way too much too fast, and we had very bad visualization of the neurons. Kind of none of the things were good. But it was our first try, so we kind of we knew that it wasn't going to work. We just kind of wanted to work through as much of the process as physically possible, so that way we kind of we've tested everything a little bit, got a little bit of a feel for it, so that way when we come back to it, we could learn from all of our mistakes instead of still having so many unknowns. And that was kind of the thinking on that. Yeah, so this time we're going to improve on all of that, hopefully. And basically the idea behind today's stream is we're going to kind of go step by step on what the new plan is, because I realized, because I tried to do too much too fast last time, I think the best plan is to basically take this project and break it up into a bunch of smaller, still very interesting pieces that can each be their own video, but that way we're not trying to rush through too much too fast, and it'll also let us experiment with some other things that we didn't get to last time. And also some, they're kind of tangential, but I think they're equally interesting, or even more so. And so we'll kind of go over this. So yeah, let's get into it. So first things first, I need a new incubator. My new lab, if you saw my new lab video, is kitted out specifically for growing microbes. It's not really meant, or it wasn't originally meant for growing mammalian cells. But really, the only thing that I'm truly missing from mammalian culture is a CO2 incubator. And a CO2 incubator is just an incubator that has a CO2 sensor and a solenoid and a gas bottle. Like, it's not rocket science to do this. And we'll be following, or we're kind of basing our design off of this tutorial, written by Andrew Pelling from Pelling Lab. He's a researcher out of the University of Ottawa. He's the one who makes the apple ears and where I got the idea for the meatberry project from. Very cool guy. I got to meet him once. Nice dude. He kind of brings that biohacker energy to academia, which is kind of fun. But either way, this is his kind of thrown together incubator. Ours is going to be a little tidier, mostly because he built this incubator out of a styrofoam box, and he was trying to do both heat and CO2. We're going to skip at least one of those steps. And oh, thank you, KZ. Greatly appreciated. Yeah, so we're gonna be skipping at least one of those steps. We're starting with an incubator just off the shelf. It's a reptile egg incubator. It's the same one that I use for all of my E. coli stuff, but it does all the heating on its own. So literally, all we're gonna do is we're gonna drill a hole in it, stick a tube for the CO2 bottle, add a solenoid, an Arduino, and a CO2 sensor. And that way we can just measure the CO2 and put in pulses of CO2 to keep it at about 5%, because that's what you need for mammalian culture. You need an incubator that can be at 37 degrees at 5% CO2. Now, the CO2 is not actually there to like replicate the conditions that the cells would normally be experiencing. Because if you think about it, like muscle tissue or neurons aren't actually in a fluid that's extremely oxygen dense, because they get their oxygen from your blood, but it kind of diffuses pretty slowly. So it's not a very oxygen-rich environment. But that's not actually what the CO2 is for. The CO2 is really there as a buffer. So one of the components of the media, and we'll talk about this in a moment, but one of the components of the media is a carbonate-based buffer. So by having CO2 in the atmosphere around the cell like culture vessel, you basically make it so that CO2 will continually dissolve into the liquid and continue to sort of re-up the buffer to hold the pH at the right level. Because if the pH swings, you'll kill the cells and then everything dies and then you're not having a good time. So nobody wants that. So this is kind of the plan for this. We're going to be using a infrared CO2 sensor, and we managed to find one from China that is able to do the CO2 range that we want, and we've already kind of got it working. It's one of those things where we basically just need to take a couple of days and just finish this project off, and I'm thinking of doing that in the next couple of weeks, so that way we can kind of start getting some of this going. So it should be fairly simple. Like we've got the code that's basically working. Essentially, all we have to do is just finish wiring it up, stick it in the incubator, and it's done. So we'll have to test it. One of the things is, how do you calibrate? Oh, hey, somebody else kicked in. Greatly appreciate it. Thank you. But yeah, so one of the other questions is, how do you calibrate the thing? Because it really does need to be 5% CO2. And so do you just take the sensor's word? Oh, somebody else kicked in. Thank you very much. Greatly appreciate it. Thank you, Victor. But anyway, so how do you calibrate the thing? And so that's where we're a little bit unsure, because we could just take the sensor's word for it. And that's probably where we're going to end up doing. But I may try and find like an off-the-shelf CO2 sensor that we can kind of stick in the incubator for a minute and just like test, make sure it's good. Because this really does need to be perfect, because if it isn't, the cells die, and cells are expensive. Ooh, Rostux kicked in. Greatly appreciated. Thank you very much. Yeah, I am trying to read the chat just through like the corner of my eye, but we'll do more kind of question-y stuff at the end. I will try and answer questions as they come through, and especially if you send a question through super chat, I'll try and answer it as quickly as I can. But I also want to. It's not like I want to try and burn through this, but you know, there's a lot to cover. But anyway. Moving right along. So that's the incubator. Once we have this unlocks not just the Neuron project. This actually unlocks a huge number of other mammalian projects that I have been just waiting to do for years. I have so much DNA in my freezer that I've slowly been accumulating over the years that are just different constructs that do different things. And a lot of them we're going to actually go through. So I'm pretty excited to tell you about that. Somebody says, Valdeg says, cells are expensive, which is messed up considering how many of each of us have. The thing is, the cells, weirdly, neurons are actually one of the less expensive, at least the type that we're going to buy, is one of the less expensive cell types, primarily because they're non-dividing. So the number of cells that you get in the tube is exactly the number of cells you will have in your final culture. They don't divide. So the companies know that you have to keep buying them from them. So unlike something like a fibroblast, which can just replicate almost infinitely, you're going to have to rebuy the cells regularly in the case of neuron experiments. So they price the cells much lower to make it much more affordable. So like the cells that we're going to be buying are about $250 a pop. And you get, I think, 1 million neurons or something like that per order. Lato kicked in. Greatly appreciated. Thank you very much. Yeah, so that's the thing. 1 second, I just got to deal with chat for a second. We have a fuckhead alert. Come on, why won't it? Oh, it won't. Okay, just hold on to your butts for a second. I just got to deal with this. Sorry, I don't have anyone modding my chat today, so I have to deal with the dickheads myself. Anyway, moving on. But yeah, so that's the plan for the incubator. So what are we going to grow first? Well, neurons are hard, so I don't think it's a good idea to start with those. I think what makes the most sense is to start with fibroblasts. And the reason for this is because they're both very easy to grow, and they're very easy to modify, and also they're not that expensive. So for those that don't know, fibroblasts they're found in a whole bunch of different cell like tissues, but they're mostly found in like skin and connective tissue, that sort of thing. They're sort of your like go-to connective cell, basically. There's others. There's like keratinocytes, but those are much harder to grow. The nice thing with fibroblasts is they are kind of aggressive because they are skin cells, so they divide very quickly, they grow very quickly, and also they're very easy to grow. Like the culture conditions for growing fibroblasts is basically just DMEM and FBS, and they just kind of grow. Like you don't need anything special, you don't need any extra growth factors, you don't need anything really, other than the basics. And also, there's a project that I've wanted to do for a very long time. There's a Japanese group that basically wondered, do you really need to buy the special food for these cells, or can you just give them like Gatorade? And which sounds ridiculous when you say it, but like when you think about it, all DMEM really is, and for those that aren't aware, DMEM is Delbico's modified eagle medium, which, as I've mentioned before, unfortunately does not contain eagles. It's named after the guy who came up with it originally. It's not like named after a specific thing. Ooh, we got a new member. Welcome to the channel. Thank you very much. But yeah, anyway, so this Japanese group wondered, could you grow cells without having to buy DMEM? And so basically what they ended up doing was they used this local Japanese energy drink called Green 𝐎𝐷𝖆𝖆, mixed with like an everyday multivitamin and a few other like off-the-shelf things, and they were actually relatively successful. Like they found out that you could sub out, I think it was like 70% of the DMEM with Green 𝐎𝐷𝖆𝖆, which I just find endlessly entertaining. But one of the other things that they found you could, well, so actually they didn't find this, but they did mention it. There's another paper where, so the two ingredients for growing fibroblasts, as I mentioned a moment ago, was DMEM and FBS, which is fetal bovine serum, or baby cow juice, as I like to call it. And it's exactly what it sounds like. You take a fetal cow out of cow, and you drain its blood out, and then sort of, centrifuge out all the cells, and then just save the serum. And this is it's really old technology. Like this is one of those things where we figured out that it works, but it's not like great technology, because again, you've got to deal with like the baby cows, which is unpleasant. And also you're always going to be dealing with the potential for contamination, because if that cow was sick, whatever they're sick with ends up in your cell culture. So buying fetal bovine serum can be very, very expensive, because the stuff that's guaranteed to be clean, they triple the price. And and even the cheap stuff is expensive. So somebody wondered, could you sub it out for something cheaper? And one of the options that apparently works surprisingly well is basically egg yolk extract. So essentially what they do is they sort of take an egg yolk and they mix it with a little bit of water and homogenize it and cook it, and then strain out all the solids. And the serum-y goop that you're left with apparently is a very reasonable supplement alternative instead of fetal bovine serum. So once we have the incubator up, I really want to try this. I really, really, really want to see if I can get fibroblasts growing with just Gatorade and egg juice. Because, I mean, A, that's hilarious. But also, if it works, that's actually kind of amazing. Because mammalian cells, one of the most expensive parts of them, other than buying the cells themselves, is the media. Like, the media is not cheap. So any mammalian experiment is just by default like expensive. And so if you could do this, if you could grow cells with Gatorade and egg juice, I mean, that's just kind of amazing. So I think that will be, I think that'd be kind of fun. And also I think it'll make a great video. I mean, just like that title, like growing skin on egg juice and Gatorade. I mean, come on, that's That's just fun. That's like all of my like old biohacker instincts just like all happy and enjoying it. So I think it'll just be fun. But okay, we have the fibroblasts growing. Now what? And the answer is we're going to modify them because, I mean, why else grow them, right? So there's a few things that we're going to be modifying them with, but I think the one that makes the most sense to start with is this mod. And basically what this is, all of these little tendrils are microtubules, and they've been tagged with GFP, or green fluorescent protein. Now, I'm using this image just because it looked the prettiest, but the construct that I have in my freezer is actually an RFP. So it's going to be, so it'll look like this, but it'll look red instead of green, basically. But one of the things you can notice, so you'll see all the little fibrils throughout the cell, but you see this big void in the middle? That'll be important in a minute. So what is a microtubule? This is a microtubule. There's two components to microtubules. Alpha tubulin and beta tubulin, and the two units come together and then they form this sort of spiraling, well, tube. One of the reasons I particularly like microtubules is because they undergo what's called catastrophe, which is my favorite, okay, it's one of my favorite bioterms ever. So basically when they grow in this spiral, but when they fall apart, because they're constantly growing and shrinking, right? So like as the cell moves around, that's actually part of how cells move, is they extend and shrink and pull their microtubules, their actin, and all kinds of stuff. But when they fall apart, you'll see that the tubulin dimers kind of line up, and when they fall apart, the individual strands kind of peel apart, sort of like a flower opening or opening a banana. Unfortunately, I don't have the picture here, but this is actually, there's actually a sort of a secret mechanism built into this that I find fascinating. Microtubules are often used to anchor different organelles. So you'll kind of have like a stick, and on the end of the stick, you'll have an organelle of some kind. And one of the ways that the microtubules are anchored, because you got to figure, how do you get a bunch of like spaghetti basically to stick to something? The actual dimers stick together pretty good up until a point. Like once the catastrophe starts and they start uncurling. Ooh, we had a couple people throw in. Thank you very much. Greatly appreciate it. But yeah, once the banana starts to open, there kind of like passes a tipping point where the ends come off first. And that's the important thing. The ends always come off first. But if you were to say make a ring and have the banana kind of come up through the ring and start undergoing catastrophe, it sort of like locks on to the ring. And so that's one of the ways that you could both, it stops the catastrophe and it also anchors whatever has the ring on it, which I think is just endlessly fascinating. So yeah, that's just a fascinating little tidbit. But yeah, that's how microtubules work, and we'll be able to visualize them in every single cell, which is going to look gorgeous. Back in university, in my cell biology lab, we got to do what's called immunofluorescent staining. So if I just go back for a second, basically what we did was we took an antibody that was targeted to tubulin. So it's an anti-tubulin antibody, and to that antibody was conjugated a fluorescent dye called FITC. I don't remember what FITC actually stands for, but either way, it lets you visualize the tubulin, and it looks gorgeous. And I really should have stuck my tubulin image on here, because we got to, like, we did the procedure, and then we got to use a very, very expensive fluorescent microscope to visualize them. And it looks awesome. So being able to see cells like this is going to be really, really cool. But here's where things get really, really cool. So we're going to have an RFP tubulin. So that means all of the tubules are going to look red. But if we had something that stuck to GFP, or not to GFP, sorry, stuck to the DNA, that means we're going to be able to visualize both the DNA and the microtubules, which is really cool, because then we can actually visualize cell division. So I have this construct. It's called D sub GFP. Now, the biologists in the crowd might actually recognize D sub. It's called, 𝐷SUP stands for damage suppressor protein, and it's a protein out of tardigrades, which are these guys. Tardigrades are some of the most radiation resistant and indestructible organisms that we know of. Like, these things have been exposed to the vacuum of space, just terrifying amounts of radiation, heat, cold, you name it. They've been dried out completely for 30 years and rehydrated, and then just kind of like wake up, start eating, do their thing. But one of the ways that they're able to be so radiation resistant is because of this protein called 𝐷SUP. And it's sort of like a DNA shield. It basically coats the DNA in this special protein and sort of holds it in place so that if anything happens to the DNA, it can sort of immediately put the DNA back in position. Like, it's kind of a scaffold. So that way, you know, if you've got like a double strand break, the DNA breaks, the DNA repair enzymes can come in and immediately stick it back together without any amount of time for it to like fall apart, like lose alignment or any of that kind of stuff. So that way the DNA stays much more intact, even in extremely high radiation environments. So I've had this construct in my freezer for a really long time. And what I want to do is I want to put it into some fibroblasts, and then I'm going to irradiate them and see if they die. Because there's a paper that did this. They took human cells, expressed 𝐷SUP in it, and just blasted them with enough radiation that like you would be stone cold dead if this happened to you. But the cells kept on living. I mean, some of them died, but the vast majority of them survived much, much better. Like, they were definitely under an enormous amount of stress. Like, they're not like happy cells after being hit with 5 Gray of radiation, but they did survive, which is, I think, a testament in and of itself to just how kind of amazing these proteins are. But because they stick to DNA, it's also a really easy way to visualize DNA in living cells. So this green image is the 𝐷SUP. This pink one is most likely an antibody. I don't remember. I'll put links to all the papers and all the stuff that I'm going to talk about today in the description box after the end of this video. But normally when you're doing immunofluorescent imaging or stuff like this, you'll use either a chemical dye called DAPI, and it will make DNA blue, like fluorescent blue. And so that's one way to visualize DNA. But the problem with this is all of these dyes you can only apply after the cells have been fixed. So basically you have to dry them out, fix them in formaldehyde. Well, you fix them in formaldehyde first, then you dry them out, pop holes in them, and then you can like dye their DNA, which isn't super helpful if you want to see the cells as they're dividing and living and doing their thing. Whereas this 𝐷SUP, it works just fine in living cells. Like, you can just see the nucleus and all the DNA in living cells, even while they're dividing. So basically, my plan is I'm going to sort of mix 2 pieces of DNA and put them both into the cells at the same time. So it's going to be the GFP 𝐷SUP and the RFP tubulin. And the result is this. So this is, I believe, a sea urchin embryo where they've done similar modifications. But you can see the blue in this case is all the microtubules, and the yellow is the DNA. So you can see the chromatin form and then undergo mitosis. And we should be able to actually see this in cells that successfully take in both pieces of DNA. So that's going to be, I think, really, really, really cool. It's one of those things, like this GIF is amazing, but imagine this in like 4K. It's going to be awesome. Now, there is a question of, okay, great, so you've modified the cells, but how are you going to see this? Because the cells don't just look like this. Like, this is fluorescent microscopy, so you really need a fluorescent microscope to be able to see this. Well, it's a good thing I have one now. Because of some of the work I do day to day, primarily on the Silk project, we needed a fluorescent microscope, and so we got one. This is a beautiful Labor Lux fluorescent microscope, and it's definitely old. It's got like a hardcore mercury lamp built into it, and I mean, it just works so well. It makes a horrifying buzzing noise when it first turns on, because, I mean, mercury lamp. But the images that it gets are just outstanding. I mean, so these are just some RFP expressing E. coli. And I mean, you can see every single little cell. The detail is incredible. So this is on Max magnification, so that way you can actually see each of the individual cells. And you're going to want to keep this image in mind for later on in the stream, because there's going to another thing we're going to talk about is going to look very similar to this, but much, much cooler. So we just got a question from D6. Can you do analysis on how we can be as cool as you? I cannot. That is a recipe, just like the KFC herbs and spices. So yeah, sorry. I'm just kidding. I'm just a gigantic nerd, and read way too many papers. But anyway, so yeah, this is the fluorescent microscope. This is another image that I took with it. These are some yeast that are expressing GFP. So, I mean, just look at them. How cute do these look? You can like see their little nuclei and some of their little organelles. It's just, I just think they're adorable. But also you can see just how inconsistent this particular gene or this particular construct is in the yeast. And it's one of the many reasons why I've actually stopped working with yeast, because they're very difficult to get a consistent result out of. As you can see here, like the variability from cell to cell is, I mean, it's for somebody who does genetic engineering, it's actually very frustrating, because what you really want to see is this, where every single cell looks exactly the same and you're getting large levels of protein expression in each cell. Whereas this is useless because half of them aren't doing their job. However, that's okay, because these are just for show. This is there's a company which will remain nameless that sells a GFP beer kit, which I don't recommend you buying because it's awful and their quality control is dog shit. But that's where I got these yeast from. These are just Saccharomyces cerevisiae, but you know, it's not a great construct, but also it's indicative of why it's so annoying to work with yeast. But yeah, so one of the things I love about this is just the way that yeast organize their nuclei and organelles and stuff kind of makes them look like the forest spirits from Princess Mononoke, which they're just adorable. They're so cute. So yeah, I thoroughly enjoy this microscope. The clarity is unbelievable. And it just lets you visualize stuff like this. So basically we'll be able to use it for visualizing the different proteins expressed in fibroblasts. All right, so we've modified the fibroblasts. We've established that we can grow mammalian cells, and we can modify them, I should say. Now what? Now we grow some neurons, because I mean that's the logical progression, right? Like once you prove that you can grow the cells, next up, neurons. Now this time we're going to start with rat neurons, because the human neurons are much more difficult to get. There are companies that sell iPSC-derived neurons. So there was a lot of questions in the last video, which I find... oh, thank you, DC. Greatly appreciate it. Ooh, Cat donated. Greatly appreciate it. Thank you, everybody. Y'all are the best. But yeah, so human neurons are bigger. I believe their growth requirements are a little bit more finicky, and rat neurons, frankly, are just cheap and easy, and they grow nicely, so it's what we're going to start with. Down the line, I would like to switch to human neurons, and the cool thing is, where I got the human neurons last time was they were iPSC-derived, which is induced pluripotent stem cells. So basically what that company does is they take a skin biopsy from a volunteer, and they treat them either with some small molecules or a genetic construct that basically causes the fibroblasts to revert into stem cells. Oh, thank you, Bud Dog. Greatly appreciate it. Yeah, so basically the nice thing with iPSC cells is that they can be turned into any cell type you want. Or, I mean, biologists argue about this endlessly, and I mean, I find the distinction to be kind of pedantic, but they're very hesitant to say that iPSCs turn into a specific cell type. They'll always say that they resemble a specific cell type, but I find this very pedantic, because like if it looks like a duck and it quacks like a duck and it does all of the same proteins as a duck, it's a fucking duck. But either way, the nice thing with iPSCs is that you can go from skin to neurons very easily. Well, I say very easily. It's an expensive process, but it's very doable. And the nice thing is this means you're not taking from primary culture, which is where we're going to be getting our rat neurons. So the rat neurons that we're going to be using are probably going to just come from Thermo Fisher, just because that's easy, and I have an account, and I can just get them. I don't need to like go through some weird company. And also they're not expensive. They're like $250 a pop, and that's not like break the bank expensive. Like if I kill a batch by accident as we're kind of going through this process and getting used to this process, it's okay if I ruin a couple of batches, which will sort of inevitably happen. So the rat neurons that we're going to be getting are what are called primary neurons, which is sort of exactly what you would think. Whereas the human neurons were iPSC derived, so they took skin, turned them into stem cells, and then turned them into neurons. Primary cells come from primary tissue, so they take a baby rat and they, cut its head out. Well, they anesthetize it, euthanize it, etc. Take its brain out, and then there's a process for disassociating the brain into the individual cells and purifying them, cleaning them up, getting them ready for customer and for growth, and then they immediately freeze them and ship them off to a customer. So the nice thing with this is there is still a little bit of dividing potential. So some of the cells that you grow will actually divide a little bit, so you'll get a couple extra cells than the exact amount that you put into the dish, which is kind of nice because then you're not limited to exactly the number that you put in the dish. But also, primary cells are a little bit easier to grow, or at the very least, these specific rat cells are fairly easy to grow. So this is the protocol that I'll be following. One of the reasons I want to follow this protocol is because the recipe isn't hard. It's just these four things. Like there's no extra growth factors, there's no nerve growth factor, brain-derived neurotrophic factor, any of that stuff. Because every time you need one of those, oh, thank you, Pocket Rat, greatly appreciated. But every time you need an extra growth factor in your medium, you instantly add like $500 to the cost of the project, which is not terrible. Like I can do that if I need to, but if I can avoid it initially, that would be good. One of the key changes that I'm going to be making in this experiment versus last time is we used a chemical called PEI as the coating in the culture vessel for growing the neurons. And I mean, this is fine, but it's not the thing you're supposed to use. There's a protein that is one of the main proteins that neurons normally use to anchor in the brain or wherever they're growing, called lamellin. And we're primarily going to be, so I'm going to actually get some, and basically all you do is you sort of take your 6-well plate or 24-well plate or whatever I'm going to be growing the neurons in, because for the first couple of times that we do this, I'm not going to try and grow them on a neuron array. I'm literally just going to grow them in a dish, just to like prove we can grow them. Basically what I want to do is get this protocol down so well that I can be entirely confident that they are going to grow no matter what I put them into, because the arrays, and we're going to talk about this later, but the arrays that I'm going to use, at least for the first couple of tries, aren't going to be see-through. So the only time that we're going to be able to actually see the neurons is at the very end, once the experiment is completely over, we've taken our readings, and we want to just like visualize them, see how they grew, this kind of thing. But we're not going to be able to like check on them, so I need to make sure that my protocol is basically perfect so that it's reliable enough that I don't need to think about it, and I don't need to be able to inspect the cells. I need to just basically take it on faith that they're growing and be able to detect their growth via their electrical signals rather than being able to look at them, which it's yeah, anyway. So yeah, that's basically the reason I want to go about this, because if I can do this without needing to like inspect the cells, that'll be really good, and if I can do this consistently, that's also going to be really good. So that's kind of the plan with the neurons. Like I say, I'm going to start growing them in just a regular culture dish, either one of the little like self-contained dishes or, wow, there's a lot of, thank you everybody for kicking in, by the way. I haven't been addressing every single person, but it's greatly appreciated. Thank you. Yeah, basically the plan is to just grow them in little culture dishes, because this way I can do a lot of experiments at a time. And I don't need to worry so much about doing it perfect, should I say. But yeah, so that's the plan with the neurons. So if we can get them growing and they grow consistently, we can see that nice axon and dendrite extension. Then the next step is going to be, okay, let's try modifying them. Or, well, rather, this is the, before we get to that, this is just another picture from the protocol that we're going to be following. This is one of the reasons why I want to follow this protocol. These are like, proof in the, pics or it didn't happen kind of thing. They were able to successfully grow the neurons. This is what they look like. They're the same rat neurons that we're going to be buying. They grow fine. The cell survivability is very good. They grow for like days and days and days. There's also questions about how do you keep neurons alive even while they're like, growing and signaling and doing their thing. And the answer is you just change the media regularly. The protocol says to change the media once a week or twice a week. I don't remember. I don't have the protocol pulled up right now. But that's actually a pretty reasonable amount of time. That's not like we're having to change it every single day. And if you did change the media every single day, you're actually more likely to kill them because you kind of want to mess with them as little as possible. I mean, they're neurons. They're not a cell type that's used to a lot of abuse. Like they're pretty delicate. And they're much more difficult to culture than fibroblasts. Like if you look at them the wrong way, they kind of die. So this is one of the reasons I want to just do this a bunch of times. I want to get the protocol perfect. I want it to be repeatable. I want it to look good. And if those criteria are met, then we can move on to the next step, which is modifying some neurons. So this is hands down one of my favorite pieces of genetic engineering. It's a system called Brainbow, and we'll go over how it works in a second. But basically what it does is it labels every single cell a different color. And this is amazing because it lets you actually trace kind of the individual axons and dendrites and stuff. Now, it's not perfect. So this was originally developed because they thought, okay, well, if we can just label every single cell a different color, then surely we can just look at all, we can just like take a picture of a slice of brain and just from that be able to but just from that, be able to like trace every single connection. Unfortunately, the resolution of optical microscopes doesn't actually allow for that. So the only way to truly trace every single connection in a slice of brain is with an electron microscope. And there's a group that's actively doing this, where they're building a system with something like 50 electron beams in it, so that way they can be scanning sufficient quantities of brain tissue to actually scan like. They're at like cubic millimeters of brain right now. They want to be able to do like centimeters or eventually a whole brain. So that way you could map every connection in an entire brain. Brainbow doesn't quite allow you to do that because it's just not, like the fidelity just isn't enough. Like the optical resolution just is too low. You might be able to get away with it with something like 2-photon microscopy, but even then, I mean, you're pushing the resolution of optical systems. However, I don't really care about seeing every connection. I just want some rainbow neurons. And the nice thing is the version of Brainbow that I have in my freezer is not actually neuron specific. It's on just a ubiquitous promoter, like a constitutive promoter that just works in every type of cells. So we're going to test it in fibroblasts first. And that'll be fun. We'll make some like rainbow skin cells. That's neat. But we're not going to jump to the neurons. So we'll try it in the fibroblasts, make sure the construct is working. If the construct is working, then we'll test it on the neurons. And if we can successfully modify the neuron cultures repeatably, I think that it's going to be, well, you'll see what we'll be able to do with it in a minute. But anyway, so this is how Brainbow works. I just want to go over this quickly because it's just, I will talk about this every chance I get because Brainbow is just so cool. Like it never stops being amazing. So basically the way this works is you've got your promoter here, and then these little triangles are what are called LOX-P sites. Well, I mean, they're called LOX sites. There's basically what it is. LOX is a specific gene sequence which works in combination with a protein called Cre recombinase, which is here. So what Cre does is it takes two LOX sites and sort of sticks them together. So in this case, the way that this construct works, this one is called auto Brainbow. So what happens is when the promoter initially runs, when the DNA is first expressed in the cells, the first thing that happens is Cre recombinase is produced. What will happen is the Cre will bind to one of these three LOX sites, and then its partner which is either the P, the 2, or the N here. Depending on which one it grabs onto, it'll either move the promoter in front of the blue, the yellow, or the red. And in the process, it cuts the Cre out of the DNA and stops this process. So that way this only happens once. So what the end result being that you'll end up with a promoter and then one of 3 fluorescent proteins that are each a different color. If you put multiple copies of this into a cell, you'll basically make it so that the selection happens more than once. So you'll end up with some combination of red, yellow, and blue. And the end result being that you end up with this sort of rainbow that you see at the bottom. And each cell will look a different color. If you only do a single copy, you'll just have cells that are one of 3 colors, which still looks very cool. It still lets you tell what each one is, but it doesn't look quite as pretty. But yeah, that's how Brainbow works. And this is just endlessly fascinating. I have wanted to play with this for so long since I first saw it. And I think just expressing it in fibroblasts to start with is going to look amazing. And there's also a whole bunch of researchers, and I don't have the images in my little presentation today, but this works in any tissue type. So I've seen some where they've expressed it in heart tissue, and so you can actually see how cardiac cells grow and divide and form these little clusters as they're growing, which I think is fascinating. But either way, that's Brainbow. I'm super excited to try this out. And I mean, if we can get this far into the project, it's just going to be amazing. Okay, so we've modified the cells. We've done Brainbow. We've got neurons growing. Now what? Calcium imaging. So one of the things is doing the electrical interactions with the neurons is very, very hard. Like truly, it's very hard. We'll get to it, we'll make it work, but it's much more difficult than something like this. What you're looking at is a zebrafish brain that's been modified to express a protein called 𝐃𝐭𝐿6F, which, biologists are shit at naming things, so of course it's going to have a horrible name, but that's what this is. What you're seeing is neurons firing. So the way that this protein works is essentially what they did was they took GFP and they split it in half, and they fused it with a protein called calmodulin. Calmodulin is a calcium-sensitive protein, and they basically took 2 halves of a GFP, calmodulin, and a calmodulin-binding peptide. So what happens is when calcium levels are high, the calmodulin-binding peptide sticks to calmodulin and sort of forces the 2 halves of the GFP into contact, and it suddenly becomes fluorescent. So when it's broken, no fluorescence. When calcium is high, fluorescence. So when you express it in something like neural tissue, you can actually see the flashes of the neurons every single time they fire. Because when a neuron fires, it releases this enormous pulse of calcium. Or there's an influx of calcium, I should say. So you can actually see the neuron fire, and that's what you're looking at. You're literally looking at a fish thinking, which is just, I don't know, what do fish think about? I don't know, but this is what it looks like. So what's kind of amazing about this is it doesn't just work in neurons. This will work in any tissue type where you have pulses of calcium, including plants. So this particular GIF comes from a gentleman named Alex Costa. I'll link to his stuff in the description after the video. But this is was basically they took that same protein and they expressed it in Arabidopsis. And you can actually see a plant thinking. Or, I mean, thinking is maybe a little overly broad, but it does basically the same thing. Like, plants use calcium to signal. It's one of the ways that plants communicate intracellularly. So you can do very, very interesting things with it. For example, somebody took that protein and put it into a Venus flytrap. So I'm going to let this loop a couple of times because, like, it's just amazing to watch this. So for a very long time, we knew that the way that Venus flytraps work is by a pulse of calcium. So Venus flytraps have 6 little hairs on the inside of the trap. And when you poke one of the hairs, it releases this big pulse of calcium, which you can now see because of this protein, which is just, it's just so amazing. Like, this is one of the reasons why I'm a biologist, because just look at how beautiful this is. something that people figured out pretty quickly is that a single tap to one of the hairs is not actually sufficient to get the trap to close. And when you think about it from the Venus flytrap's perspective, this is probably a good thing, because if something just bumps the trap, that's not necessarily indicative of it has caught food. Whereas if it caught like a fly or some other thing, like an insect, like something it's trying to eat, it's going to be moving around. Whereas if it's just a stick or a piece of debris, it might only knock the thing once. So this way, it's basically a calcium-based motion sensor that is level dependent. So if you watch this GIF really carefully, you'll notice that in the first stimulation, there's that big calcium pulse, and then it immediately starts to fall off. But if you watch, when it hits it again, the color gets much brighter. Like, you can actually see the calcium level that is sufficient to trigger the trap to close, which, I mean, is just breathtaking. Like, that's so cool. I used to grow Venus flytraps, and they're one of my favorite plants. They're just so cool. But there's actually something that I find amazing is somebody did a study on Venus flytraps. Thank you to whoever just kicked in. I don't see the notification yet, but thank you. Greatly appreciate it. But they actually did a study where if you take propofol, which is a general anesthetic, and you give it to Venus flytraps, they don't close anymore. Like, just let that sink in for a second. A general anesthetic works on fucking plants. Like, that's just... It just broke my brain a little bit when I first heard that. And, like, if you were to combine that experiment with this experiment, which they did, by the way. Unfortunately, I don't have the GIF to show you, but they did that. Like, they numbed, essentially, a Venus flytrap, and then they poked it, and you can see the calcium wave, but they don't move. Like, it's just mind-boggling that works. Like, why? Yeah, here, I'm gonna reduce the donation sound a little bit because people are complaining. But yeah, apparently plants can get high. Wasn't expecting that, but you know, somebody figured that out. Which, again, I mean, props to whichever researcher came up with that grant proposal. One of the other fun things that I really like about this image is if you look carefully, on the surface of the trap, you'll see how it looks kind of stippled with these little black dots. It's a little harder to see because this isn't like a super high resolution video or anything. But those dots are the enzyme secreting cells. Like, so back when I was back in university, I took a plant class where we did a lot of plant sectioning, where you cut single cell thick sections of plant tissue and then look at it under a microscope and stain it and stuff. And you can actually see these like little globule things that are the enzyme secreting structures in the plant, which is just fascinating. But what I found really cool was that on the back side of the plant, like what is the underside of the trap, you can actually see the same enzyme secreting pods, but they're shriveled. So whatever mechanism the plant uses to trigger the generation of these enzyme secreting organs, for lack of a better word, they do it on both sides. Like, it's not side specific, but there's some mechanism to shrink the ones on the back so that they become inoperable, but the ones that are inside the trap stay operable. I don't know how that works. It's just amazing, and it just never ceases to be amazing. But anyway, all right, let's move on. There's one more thing that this works in: bacteria. So these are E. coli, and it might be a little hard to see. You might gonna have to watch this for a little bit, but if you look around, you'll see that there's these little pulses. So they express the same protein in E. coli, and sure enough, bacteria do the same sort of calcium pulses that we would normally only expect out of, like, eukaryotic cells. Like, so what's amazing about this is that whatever mechanism life used to make neurons must be so old that it existed in bacteria, and it continues to exist in bacteria. And the paper that this is from theorized that the calcium response is actually a way of doing mechanosensing. So it's a way for the bacteria to basically detect what's going on in their environment, which, I mean, just think about that for a second. Like, the bacteria are thinking. Like, it's not thinking in the way that we experience, but it is a thought process of sorts. Like, it is analyzing its environment, making decisions, and changing its behavior That's crazy to me. So what did I do? I bought this DNA, or I should say I found the code for it, and I had it ordered. And the company is presently synthesizing the DNA. So this is the one I designed. This is one off of Addgene. I chose these two just so you can see the two different proteins. I mentioned the 𝐎𝐫𝐢𝐠𝐴 thing before. So this is what I'm talking about. This is the 𝐎𝐫𝐢𝐠𝐴 binding peptide here. This is one half of the GFP, a little spacer, and then another half of the GFP, and then the 𝐎𝐫𝐢𝐠𝐴. So when this is working, this end sticks to this end, and it brings these two halves into contact. It's not annotated on this side because I just copied the code directly, and I didn't take the time to like annotate it carefully. But there's actually two different proteins that I had made. The first one is the 𝐃𝐚𝐿6F, which is the green one, which is what you've been seeing. But another group developed one called 𝐎𝐫𝐢𝐠𝐴, which is it's exactly the same idea. It's just using a red fluorescent protein instead of a green fluorescent protein. And so I've ordered two of these. So one with 𝐎𝐫𝐢𝐠𝐴 and one with 𝐃𝐚𝐿6F. So that way I've got a red and a green calcium sensitive bacterial sensor. And they're both His tagged. So it means that I can actually grow this in bulk, extract it, and I can actually do the calcium sensitive thing just like in a test tube. So theoretically what I should see is I'll have my test tube of what should look like clear liquid. And then then if I've got a UV light shining on it, you shouldn't see anything. But if I drop a little pellet of like calcium chloride into the solution, everywhere the calcium touches should immediately start glowing, which I think is going to look amazing. But also it's just a really cool way of being able to show this amazing, amazing piece of bioengineering without having to necessarily, do it in neurons. But once I've got this working in bacteria and we've modified the neurons and we've got them growing and all this kind of stuff, absolutely I'm going to be doing this into the neurons. So that way, rather than having to necessarily connect electronics to the neurons to see them working, I can just take a picture. And basically what I'll do is I'm just going to essentially jam the microscope objective into the solution, and we're just going to watch the neurons fire for a little bit. And I may end up, depending on how this goes, I may end up doing this to the ones that are on the final electrode arrays, but I'm kind of undecided on this, for lack of a better word. Yeah, so that's the plan with the calcium imaging, and I think it's going to be gorgeous. I think it's going to be amazing, and I'm so excited to try this out. And this construct should show up in a couple of weeks, so I'll be able to hopefully get going on this. I know there have been a lot of complaints that I haven't been making a lot of videos, but the reality is I'm just really busy. My day-to-day is actually focused on the Silk project, and it has taken a huge amount of effort, and kind of it's been taking some adjustment to sort of figure out that like balance between the work stuff and also making videos. Not that making videos isn't also my full-time job, but I've finally been able to find a balance where I can actually do both, where I can work on the Silk project, I can work on the video stuff, and so I'm going to be churning out a lot more videos soon. I've also been changing how I'm doing videos, so there's going to be a lot more streams, there's going to be a lot more short content, and I'm also trying to pick videos that are easier wins, for lack of a better word, just to kind of like get the wheels turning. But also I don't want to put stuff out until videos are fantastic. And that's part of the other reason why there hasn't been content. It's not that I'm not working on them. It's that I refuse to put out garbage, and I really want the next videos that I put out to be the best I've ever made, and I'm going to hold myself to that. So that's just a little bit of a tangent, a little bit of explanation of like what's going on with that. But yeah, so yeah, I know. So Silk project, unexpected update. I know. Somebody mentioned so I commented on Smarter Every Day's new Firefly video that I'm working on a bioluminescence project, and I'm not going to tell you about it yet, but I am actively working on it, and it is stunning. Like, it is literally some of the prettiest footage I've ever gotten, and you guys are going to like lose it when you see this. I'm not going to talk about it yet. Can't tell you what the project is, but I will tell you that you're going to love it. So anyway, that's kind of that. So let's keep moving. Okay, so we've done the calcium imaging, we've done brain boat, we've got neurons growing. At that point, oh, actually, sorry. Okay, let's do one more GIF. One more GIF. I can't resist. So this is the two different proteins, the 𝐎𝐫𝐢𝐠𝐢𝐳 and 𝐺CAMP6. This paper expressed each in a different neuron type, so you can actually see the difference between the different neurons as they fire, which I think is really cool. And this is done in a mouse brain, where they're like showing pictures to a mouse, and that's what that little arrow indicates. Is they show the picture and then you see the pulse, which I think is really fascinating. Sorry, let me just get fucked. Oh, there you go. Anyway, so we've talked about the modifications. We've got the neurons growing. Now it's time to actually do the electronics. And yeah, this has been made infinitely easier by this company called 𝐎𝐵𝐷 Technologies. They've actually built a series of chips specifically for interacting with neurons. So before I tell you about this, I got a couple of comments I just want to answer really quickly. Markov says, looking forward to the projects. Thank you for the donation. Greatly appreciated. Guentarino says, could you use machine vision to take input from the neurons? Like if a particular area lights up, would that be interested as an output? And the answer is yes. So there's a fantastic researcher, Professor James Potter, who sort of started the field of multi-electrode array neural recordings. And as far as I know, he's a subscriber. Like he might be watching this, and like if he is, say hi in the chat. That would be dope. But one of the ways that he interacted with his neuron arrays was he had the electronics. So he was electrically monitoring the neurons, but he was also using what's called two-photon microscopy to see the neurons firing. So that way you could kind of correlate the visual data and the electrical data. And if I could do that, that would be amazing. But for our initial batches of electronics, I just don't think it's going to be possible. But yeah, I mean, theoretically, yes, you could absolutely interact with the neurons this way, where the outputs are solely visual and the inputs could be solely electrical. So you could be sending in pulses and just looking for the response. The alternative is you could do this entirely optically. So you could do the 𝐺CAMP6 for the visual, like seeing the neurons fire. But if you wanted to be able to interact with the neurons, you could also express something like, I want to say it's Cry4, but there's an optogenetic system where you basically express this protein in the neurons and they become optically sensitive to blue light. So when you shine blue light on the neurons, they immediately fire. So this could be one way of sort of interacting with them, where you could shine a very dim pointed laser at a small portion of your culture medium and use that to pulse the neurons. So that way you could essentially send in pulses of blue light and train the neurons to fire and produce a specific pattern when you're shining the blue light on them. And so that is actually something I've considered. Keep in mind, this is like 6 videos down the line. Maybe we'll do this. I don't know. But yeah, no, it's totally reasonable and totally possible. So yeah, anyway, so let's get back to the electrical side of things. So assuming we don't do the optical system and assuming we go with the electrical system instead, this is what we're going to be using. So they have a few different varieties, and it's hard to overstate just how cool this is. So this is what the chip looks like, and what I think is amazing about this company is that they actually sell the bare IC. And I believe the IC itself is open source. So what's amazing about this is if you were so inclined, and let's say you're a billionaire fuckhead who likes torturing monkeys, you could build a brain implant using this chip. So you could basically take this IC design and basically take the design from something like a Raspberry Pi and all of the other onboard components that you would need and build it all onto one IC and make a brain implant out of this thing. And you can see how tiny it is. It's absolutely minuscule, and yet, which would make it, perfect implant material. But anyway, what this company does is they offer two different series of chips. So one set is read-only, so it only detects pulses from neurons, or whatever other electrophysiology stuff that you want to do. And the alternative is they have one that also does stimulation. So that way it's both a like you can both record from the neurons and stimulate them all with one chip without needing extra hardware, which I just think is amazing. Each of the chips also come in a variety of different sizes and like channel sizes. So I think the lowest they sell is 16 channels. They've got 16, 32, 64, 128, and 200. I'm bad at binary, whatever the next number up is. So yeah, basically, like, this solves so many of our problems. Like, this is so much better than the Spyker box. This is an IC specifically designed to do exactly what we want. And, you know, if I wanted to spend like big money, I could go with their backend, because this is just what's called the head stage. So this does the actual like neural recordings, and then it pipes all of that data down a data line. But to be able to power this and interpret that data, you need an extra piece of hardware, which, like I say, I could just shell out for the thing that this company makes, but it's pretty expensive. So, and yes, I am referring to Neuralink. Thank you, 256. I'm bad at math. Okay, I have turned down the donation noise. Hopefully it's quieter. Yeah, anyway. So yeah, the head stage on their own is pretty expensive. I think it's like $1,000 for this little chip. But I mean, worth it, because it saves so much effort. Like, I don't need to like design anything, no extra components. It just works. So that's lovely. But you still need a backstage for actually interacting with this thing. And of course, the more channels you have, the more beefy your backstage needs to be to interact with the head stage. So yeah, this moves the project forward just oodles and oodles. However, if I don't want to spend the money on theirs thing, I could build my own backstage. And so this paper, Lightweight Electrophysiology Hardware and Software Platform for Cloud-Based Neural Recording Experiments, is written by these amazing people. And what they've done is they've built a backstage specifically for interacting with this chip. So it's called the Braingineers RHD controller, built based on a Raspberry Pi. So this makes interacting with this thing so easy. Like, you literally just give it the supply voltage, connect it up, no fuss, no muss. It just works. And I mean, that's really what I love to see. So this is what we're going to be using. And I've actually ordered all of the components to build this already, because, you know, chip shortage, figured it was probably a good idea to get this in advance. But with this, a data line and the head stage, we're like 90% of the way through the project. And all we really need at that point is software to interact with it and the arrays themselves. I'm still contemplating buying an off-the-shelf array, and we'll get to that in a minute. But even if I make the array myself, just having this simplifies everything so much. But yeah, I mean, as people are mentioning in the comments, the signal processing on this is going to be an absolute nightmare. So when I was first kind of really trying to give this project another go and really starting to dig into the research, I was contemplating opening it up and like starting a whole Discord channel just for this project. And I may still do that. I'm still undecided, but I really, as I looked into it more and as I started finding these things, it really felt like it's still something that I want to try myself. Not because it's like I've got a giant ego and I like require doing everything myself and I don't want any help, but just because it's just fun. And I want to see how far I can get through this project basically on my own, or I should say with my team, because I don't work alone these days. I've got a team of wonderful people. Sorry, I gotta. But yeah, so, you know, with myself and my team, I think we can handle most of this. I think where we're going to get stuck is in two places. One is the actual array design, but I think we can actually handle that now. And then the other is going to be the software. Conveniently, and the reason I've got this pulled up is because the same people who made that fantastic backstage that we just talked about a moment ago are BAMFs and posted not only all of their code, but all of their schematics, all of their like shopping list, all of their software, everything, all on GitHub. And I mean, I just, you know, we love to see it. It's just amazing when people share their work properly. So we're going to be following their stuff as closely as possible because frankly, they're awesome. And I like, props to these guys. Go check them out. I mean, they're amazing. I don't know if these guys are even still in university. The paper was a couple years old, but either way, I hope them success in anything they do because this is just awesome open source science, and I love it so much. So yeah, we're going to be following their stuff. It simplifies everything so much. But even if we get to this point, we've made the array, like we make arrays, we've got all the hardware done, we've got the neurons growing, we're using their software, that still doesn't finish the project, right? Like, okay, great. Now we're just going to have recordings of all the neurons. We're still going to need a piece of software to take those recordings live and do something with that data. And that's probably where I'm going to ask for some help. So once we get that far into the project, I may start a Discord channel or something that'll just be open for anybody who wants to help. So that way we can get some really cool software to make this do a variety of different things. But, you know, we'll see how far I can get with my team, but we'll kind of cross that bridge when we get there. Okay, last up, new arrays. So I've already started working on this, but one of the things that kind of made my decision for me, and I say that, but I'm not actually 100% like committed to this plan yet. But one of the things that is guiding my decision is the fact that this exists. So there's a company that sells multi-well plates. So it's, you know, a standard 24 well, 64 well, or 96 well, I should say, plate, where each individual well is an electrode array. And they come in a variety of different electrode densities and electrode diameters and spacings and all this kind of stuff. But at the end of the day, it's just made out of PCB material with gold-coated contacts. Like, it's not a special material. It's not glass. It's not titanium. It's just PCB, copper, gold on top. And that seems to be more than sufficient for doing neural recordings. And if the neurons can grow on that, I mean, then why wouldn't I use it? So I think my plan, at least for the beginning, is I'm going to, with the help of either some friends or maybe you guys, depending on how this goes, is we're going to design some new arrays, and we're going to just have them made by a traditional PCB manufacturer. Then we'll clean them really well, make little like retainer rings to go around each individual set of electrodes, and then just turn them into culture dishes. One of the things that I'm going to be changing, because I mean, the nice thing with PCB is they are also heat tolerant. So I could just autoclave this, which would be amazing. Like if I could just order some PCB material and then, did someone just kick in $120? Thank you. Thank you so much. It's greatly appreciated. Yeah, I've missed a few of the donations because I turned the donation sound down, so I haven't been hearing them. So thank you everybody who's kicked in. It really is greatly appreciated. And wow, we're like getting close to our goal. This is based as hell. Y'all are amazing. Did I get a Discord? What? Oh, I did get a Discord notification. And also, I did not turn down the thing. That's my bad. Now it's turned down. It's 6969. That's hilarious. Anyway, but yeah, so, you know, if I can just make arrays out of standard PCB material using standard PCB techniques and basically off-the-shelf materials, this simplifies everything. There is still the option that I could make them on glass. We did build our new magnetron system, which works beautifully. So I could make arrays myself if I get into some like lithography stuff, which I may still do just because it's fun. But I think for getting going on this project and to like, if the goal is to succeed and not just fuck around, I think the best option is to just go with PCB based arrays because it's easy, it's cheap. And also we can just share the files. And also I could have like normally if you order PCBs, you've kind of have to use up the whole plate, right? So I could get like 80 arrays for something like $120 or $200. which is so many different experiments that we can try over and over and over again. I could even, sell extras on my website or something if I really wanted, which could make it like, if we get to the point where we have working arrays, growing neurons, the protocol working, everything working, I mean, I'm not going to sell a kit, but just having the information out there and having the arrays available for cheap, I think would allow a lot more people to play around with this, which I think would be awesome. Yeah, so that's sort of the plan with the arrays. Like I say, we do have the new magnetron, so if I wanted, I could go with glass arrays so that I could actually see the neurons. And again, I may, most likely, I'll probably make a couple. And I'm actually working on a few different lithography techniques. I'd really love to make like a laser stepper. It's the same that like Sam Zaloof or what's his name, Hugin Optics has made. And I mean, for all I know, I might be able to just pair up with them and do that. I know that I've talked with Ben from Applied Science, and we've talked more about him potentially making some arrays for me, and that's still an option. So I might go that route. Like for the video, it might make sense to just make a couple of arrays on glass just for the sake of the visualization. But I think for the sake of the experiment succeeding, I don't actually think it's necessary. Like, I don't think it's mandatory if our protocols are just tight enough that we can just reliably culture neurons without having to think about it too much. Of course, it's nice to be able to see the thing, but we'll see. The other option would be to, rather than doing PCB material, is we could use Kapton, because Kapton is actually semi-transparent, translucent. So we might actually be able to like see up through the arrays while the neurons are in there, rather than just at the end of the experiment. So again, it's an option. So silver instead of gold. Gold is more inert, so gold is better. And also the amount of gold we're talking about here is like nanograms. Like it's not an expensive amount of gold, which I realize how ridiculous that sounds, but it's true. Gold is not actually expensive if it's just a coating. It's expensive when you need like a gram or an ounce or something. But when you're talking like milligrams or micrograms, it's not an expensive material. And it works really well. So this is one of the array designs that I've been tinkering with. It's not perfect. The arrays are still... this was one that I was just kind of playing with because I wanted to see if my 3D printer had the resolution required to use as a lithography setup. Because I thought, hey, wouldn't that be fun if my 3D printer, which has 30 micron resolution, was able to expose photoresist, because then I could just make the arrays in-house. The reality is it didn't work so well. So like that's how this was made. You can see that there's a lot of failure on the photoresist, so it's not a perfect solution by any means. So I think it just makes the most sense to just have it done professionally. But I figured I had the materials, I might as well give it a try once. But there's only so much you can do with it. Yeah, silver is more conductive, but at the scales we're talking about, it's not a big deal. And no, you don't buy nanograms of gold. You just pay the company to coat the thing in gold, and they buy like grams of gold and dissolve it and make coating solutions. Like that's just, their coating solutions can be reused many times, so they're able to sell it to many customers efficiently without having to charge an arm and a leg. But yeah, so this is the design that I tried. Didn't work so good. It also actually has too many electrodes for what I'm planning. I'm thinking for the very first round of this, probably 32 electrodes is sufficient. Like, of course, I'd like more. I would love to be able to have a 256 electrode array, but starting with that is, well, it's a nightmare. It's so much data to be dealing with at once and like be pushing down a USB line that it's just not feasible as a starting thing. So I figure 32 electrodes is probably a reasonable amount, because with 32 electrodes, I could do 32 sensing electrodes and like 10 or 5 like stimulation electrodes. So that way we can actually like talk to the arrays, which is where we get to this. Okay, let's say that we get to this point. We've got the neurons growing, we've got the electrodes made, we've got the electrode arrays made, I should say. We're getting the signals out, we're able to stimulate them. Now what? What do we do? Well, I mean, like I said at the beginning of the stream, I want to make them pass some butter because, I mean, duh, that would be awesome. But so one of the papers that I believe it was one of the papers that Professor James Potter wrote, but don't quote me on that because I don't know if that's true. I don't have the paper pulled up right now. But one of the ways that they found you could interact with the neurons was sort of as when they get a stimulus, you train them to produce spikes accordingly. Oh, thank you. I can't pronounce it. Snazzy Bob? Thank you. Greatly appreciated. So basically, you can train them to respond to specific sets of signals to specific areas on the array. So like if you divide the array into little sections, you can train the neurons that like if you stimulate this little section, you're not going to turn the stimulation off until they respond a certain way. So you kind of get this like call and response thing going. And we figure that it's probably reasonable to be able to get 2 call and response signals in and out from the array. So the way that I'm thinking that this would work is it would kind of actually be sort of like a cyborg computer. So you have the computer doing a lot of the work and then the neurons doing a very small amount of the work, but they're still doing some of the work. So they're like in the loop. So the way that I think this would work is you would have a computer doing speech to text and then parsing, looking for specific phrases. So if you were to say something like, open the pod bay doors, Hal, you could have the program parse that, recognize its phrase, and when it produces the correct spike pattern, it triggers the computer to play a message that corresponds to that spike pattern. So something along the lines of, I'm sorry, Dave, I can't do that. And then if you said a second phrase, which the computer could look for, like, why not, Hal? You could probe a different part of the array and look for a different spiking pattern that would then trigger the computer to go, you know why, Dave? Or you could do this with anything. Like the nice thing is you could just set the computer to recognize whatever phrase you want without having to retrain the array. So you could do something along the lines of, pass the butter. What is my purpose? You pass butter. Oh my God. And like you could have it so that like when you say like pass the butter, you could make a little robot move, drag some butter, and then have it like tilt and look up at you and then play the clip that just goes, what is my purpose? And then you go, you pass butter. And then it just, make it just do the animatronics such that it does, sorry, I'm going to just mute my phone. So that it does the thing. Like you can basically like make the little robot do the little thing, and the neurons will be technically in the loop, but it's not like they're doing most of the work. And I think that's just going to be based on sort of the limits of what we're doing. Like we're only working with 32 electrodes or 64 electrodes. Like it's not the extremely dense arrays that you would really need to be able to really like feed them in a whole last sentence and have them understand it. So there are some researchers who are now at arrays that are like tens of thousands of electrodes dense, or they've basically built like a CMOS chip where they've basically like changed the architecture. So instead of it looking for photons, it's literally looking for neuron pulses. So you've got like a normal-ish CMOS sensor that's being able to like interact with and record from thousands of neurons at extremely high fidelity. We're not doing that. I'm not that good, and that would be way too expensive and would take like the rest of my life. Or rather, I would say I'd have to like that would have to be the only project I do, and I'm not that committed. But I think it would be reasonable to be able to get at least 2 call and response things out of a little simple neuron array. Because, I mean, it doesn't need to be perfect. It just needs to have thresholds and pattern recognition just enough for the video. Like the whole point of this is to go through the process and show how this works and like show this cutting edge neuroscience without necessarily having to like become a neuroscientist and this have to be my job, if that makes sense. I want to be able to demonstrate this, but I also don't want this to be the only thing I do for the rest of my life. But the other thing I would really love to be able to do is if you're able to do call and response like that, then it's actually much easier to do something like this where you're flying, like you have the arrays controlling the flight of a plane in a simulation. Because really all you need the robot to do, or the robot, the right? caught myself there. The neurons to do is you basically just need it to be able to control like the pitch and yaw of a plane, and you could basically just have it so that when the pitch is wrong, you're sending in high frequency noise essentially into two spots of the array depending on which way it's pitching or yawing, and same with pitch. And so you really only actually need 4 inputs, and you can essentially control a plane. This is why the papers where they did this is a little bit of a hack, because you don't actually need that much complexity to do this. It's still amazing that you can get a neuron array just even to control that. But it's not like you're able to like, give it a flight plan and go, go to Mexico and then turn left at wherever. Like that's not going to happen. But if you're just having on a self-balancing system where it's able to just kind of like keep a plane flat, that's actually relatively easy, all things considered. I mean, we're talking about growing neurons in an electrode array. So, I mean, how easy is it really? But it's, roll. I'm bad at planes. I don't fly. Oh yeah, I guess this is yaw. So I guess, yeah, roll and pitch. I don't fly, as you can tell. Yeah, so that's probably the extent of what we're going to be able to do with this project. I don't think we're going to be able to push it much further than that. I mean, I could be wrong. But I think if we get to that stage, I think that's impressive enough. I think like if you can make a robot pass butter with actual neurons, I think you just win the internet. So like that, I feel like to me that feels sufficient to be like, okay, the project is done. But I mean, if we get to that stage, I mean, of course, I'm planning to open source all of this because like there's no business in this for me other than the fact that it's awesome and I want to share it. So I think it's more than sufficient to just open source it at that point. And as we get through the project, I may just start that Discord channel and just kind of let the project kind of have a bit of a life of its own, so that if people want to play with this, we'll have the technology and we'll have the equipment to be able to just run an experiment. So like if people write new software or they come up with something really clever or a new thing you can do with the arrays, like I'd be open to running that experiment once we do the thing. But I want to get to this point first, because I mean, realistically, this is already probably blue sky that I can even make it this far, because this is ridiculously difficult. Like I'm talking about this like it's not ungodly difficult to make all of this work, but it is. But that's also why I'm going to try and break it up into pieces. So I think the first video will probably be, growing the fibroblasts and modifying them. The next one will be growing neurons and modifying them. And then I'll do just a whole video on the calcium imaging. And then the next one will be building the electronics, getting it to work. And then the last one will be, put it all together and like show it off and this kind of thing. But yeah, so, technically, there's a lot of people saying that, if you could do this, you could make a general intelligence or you could make a AI or whatever. I mean, yeah, technically it's not gonna be very good. But there's a couple of companies now that are really focusing very hard on this and, are able to probably go a lot further than we are. Because again, this is a YouTube channel. We are not a neuroscience company. I'm doing this for fun because it's awesome, not because I like want this to, be my thing. So there's a lot of companies that are building much better sensors than what we're going to be using, but I think it's still going to be valuable enough to like show the technology and show how it works and let people really see how this stuff works and how amazing it is. And that if we could get through this, I mean, that's like the pinnacle of like DIY biology. I feel like you've grown neurons, modified them, stuck them on an electrode array, interacted with them, had them fly a plane. Like how much more do you really want? Like I can't make cat girls. Like that's too hard. This is easier. And yeah, I know some people, I'm doing this for fun. And then we find ourselves in psychopaths. Look, the thing is, what I'm doing is just not going to be like that scary, I feel. Like the implications of this are definitely terrifying if you were to increase the size of the array. Because, I mean, keep in mind, we're growing something that's going to be this big. If you were to take a slab the size of, say, a pizza box, and that was all electrodes and neurons, yeah, you're now dealing with a very significant amount of intelligence. It's going to be weird, and it's not going to be anything like what we're used to, because it's not going to have the structures built in the way that like normal human brains do. So that would be fascinating, and like that actually starts getting into like some ethical stuff. But at the scales that we're at, we're talking about like a couple 100,000 neurons. Like it's the equivalent of a fruit fly. Like I've done the math. 100 like a fruit fly has, or a bee, like a single bee. Like nobody cares what happens to a single bee. Like that's the level of neurons we're talking about here. It's not like a thing that's going to be conscious, especially because it's going to be completely disorganized. Like there's not going to be structure the way that you would get in a brain, and that's so important for things like consciousness and being able to actually like really understand your world. Like this is something that is it's pretty stripped down. It's disorganized. Like it's literally able to spike on command. That's sort of it. It also doesn't have any sensory neurons. Like there's no way for it to interact with the world. So it's not like it's getting like sensory input or pain or any of these kinds of things. And like, so I think it's a reasonable spot to stop, because any further than that, and now we're actually getting into like ethical stuff where like I'd actually have to put some thought into if I feel like this is okay. But yeah, like I'm more worried about like computer-based AI than I am about like 100,000 neurons in a dish. Because you can do a lot more with 100,000 computer neurons than you can with 100,000 meat neurons. Ironically, you can do a lot more with meat neurons than computer neurons presently, but I mean, that's only when you get into the billions. But yeah, so that's the plan. So yeah, we have come to the end of my little presentation here. So I'm gonna take some time to like answer some questions, chat with you guys. But yeah, that's my plan. That's how this project is going to unfold over the next several months. I am actually starting to push this forward. I'm starting to grow my video team so that way I can be producing more videos and more stuff and be pushing this project and other projects along further. But yeah, so what do you guys think? Also, I'm going to just take a second to just go through. I've missed a ton of the donations. You guys are awesome. Thank you so much. It's really, really, really fantastic. Somebody said they prefer gold on titanium over gold on silver because it's more compatible and less cytotoxic. I agree, but it's going to be gold on copper just because, like I say, I'm going to be going through traditional PCB manufacturing for the arrays, so that's sort of my only option. I grew my own video team. No, I'm hiring people for my video team. I didn't like grow them in a tank. The tank isn't finished. Okay, so questions about how you train them. So in the literature, the way that they're trained is they found that certain frequencies of electrical stimulation are, like the neurons will respond differently. So lower frequencies tend to reduce spiking, whereas higher frequencies tend to increase spiking. Or I might have that backwards. I'm going off memory here. But it's the sort of thing where you can train them to produce lots of spikes in specific areas by basically feeding in like higher low frequency electrical noise into specific spots on the array and training them that the noise only stops when they spike in a specific spot. So like only when do you see a spike pattern in a specific quadrant do you turn the noise off, and that's how the neurons come to know that, oh, okay, that's what we need to do to get this annoying thing to stop. So it's a bit weird. One of the other things I really want to try if we get this far is to play music into the arrays. There've been a few people or a few groups, I should say, where they took a neuron array like what we're planning to do, and they fed music into the arrays, just like as electrical noise, basically, and then had and basically correlated spikes on certain electrodes to different instruments. So they had the neurons playing instruments based on or in concert with a musician playing music into the array. And it sounded very weird, and it's definitely art. It's not like real science, but it's neat. I'd like to see what would happen. Do you think ethics halt the development of cooler science projects? No. Especially when we're talking about like academic labs, they get away with just the craziest shit. And it's a little scary, honestly. Like I'm far more afraid of academic scientists than I am of like biohackers, because biohackers, I mean, most of the time are just like fucking around and don't have enough supplies to do any damage. Whereas an academic lab with like, $1 million worth of grant funding can do all kinds of crazy stuff. Like there was a lab that printed out horsepox just because they could and just to prove that they could. and by ordering like the bits of DNA from a bunch of different companies. So, yeah, ethics boards will approve all kinds of crazy stuff. I mean, look at what they do in animal labs. You know, it's an ethics board ultimately is just a group of people, and if you can convince them that your thing is a good idea, they'll let you do it. It's not like magic. It's not like a bunch of like ethics professors sitting there going, you know, should we do this? It's just people, and most of the time it's their colleagues. So it's like, you know, anyway, they can do all kinds of stuff. I like to hold myself to some amount of like ethics, so this is why I'm calling it at like 100,000 neurons. Like I'm not going, or 1,000,000 neurons. Like I'm not going further than that, because that now we're getting into a thing that like I don't know if it could feel pain, but like it might I don't know. I don't feel like crossing that line, and I don't want to know. So that's kind of where I'm going for. Alrighty. What else we got? How much local control are you going to have over this system? None. That's the trick. The array trains itself. So you're just going off of basically when the thing does the thing you want, you stop stimulating it. So it's going to train itself. It's going to figure out the weights and connections, and it'll figure out something that works. So that's kind of nice. Do you ever think that you would make something like a neural latch, something that receives an input that you can then read that input back later? Maybe, but I don't know. I mean, what might be kind of fun, and something I've considered, is what happens if you connect two arrays to each other? Like if you've got, you know, two arrays next to each other, what happens if you connect them? Or like what happens if you were to take, I don't know, like a neural recording from like a person and feed that into the array? I have no idea. It might be fun. I don't know what kind of like usable anything you would get from that, but you might get something. It could be interesting. It says, what kind of creature are we talking about with 100,000 neurons? A bee. Like a large fly. Like something that, you know, nobody really cares about so much. How are you planning to control contamination of the cells, i.e. neurons? Penicillin, streptomycin. If we, here, I'll go back on the thing to where yeah, here we go. So this is the growing step that I talked about near the beginning of the stream. One of the things you'll notice in the growth media is 1% penicillin streptomycin. So not only is this will basically prevent any bacteria or any of that other stuff that gets in there from growing, but also all of the culturing is going to be done in a flow hood. I have a proper like laminar flow hood now, so I can actually do very sterile cultures. I have to for my work. So sterility is just the base state of biology. So that's controlling contamination isn't something I'm too worried about. But yeah, I'm going to go all the way back to the end. Boop. All right, there's the end. Okay, I'm going to just go through some of the other donations, see if there were any other questions that I might have missed. I'm not seeing too many. And again, thank you everybody for kicking in. It really is greatly appreciated. Okay, somebody said, so Philip asked, is it possible to map neuronal connections using videos of, oh no, wait, I answered this already. Yeah, using optical microscopy. Yeah, if you're doing the 𝐔𝐾AMP stuff, yes, you can. Can bugs feel pain? Yeah, probably. I mean, almost certainly. Like there's a bunch of neuron studies where they'll train insects to respond to like a pain stimulus, where they basically shock them. They'll like turn a green light on and then shock them, and then see what they do when you turn the green light on, even if you don't shock them. So I mean, they definitely don't like it. Okay, what else we got? Okay, question about the plants. Do you know any studies researching the electrical impulses of plants and if they can communicate with mycorrhizal fungi in the same way? Yeah, no, there are definitely studies on this, and it seems like, yeah, they do. Also, the 𝐔𝐾AMP6 thing, I'm going to go back to it for a second. So somebody took this protein and expressed it in fungi. And yeah, fungi do this too. And it looks a whole lot more like this than it does like this. So that's pretty weird. Also, there was a bunch of electrophysiology studies where they basically just stuck a bunch of electrodes into different mushrooms, and you could do a thing where if you had like a block of mushrooms and a bunch of like mushrooms fruiting at the top. If you stick an electrode into one of the mushrooms and then like hold a flame up to a nearby mushroom, you'll actually detect what looks a whole lot like an electrical, like an action potential in the other neurons. Like they're neurons, the mushrooms. Like they respond to damage and stuff, and they produce things that look a whole lot like action potentials. They're much slower because, I mean, it's a mushroom. It doesn't need to live fast. You know, they're practically immortal when they're at the scale of some of the you know, colonies that are all just one giant organism. But their action potentials are much shorter, or not shorter, longer. So they take place over like minutes. But it's still, you know, nice big pulse and then drop, and then nice big pulse and then drop, and like depending on what you do to the mushrooms, you can actually get like shorter pulses that are faster, which I think is just strange. Yeah, somebody made mushroom music. I've seen that video. It's wild. Okay, what else we got? Oh yeah, there's a paper on. So somebody says, I saw in popular media that suggested that fungal networks seem to have language-like communication. Know anything about that? Yeah, I read the paper. It's pretty weird. It is distinctly weird. Like they found that those sort of action potential like things seem to follow specific patterns, and they could find specific patterns that always seem to follow in a specific pattern. So you'd get like a specific spike pattern and then a pause and then a different spike pattern, and they always seem to happen in a certain order. And like depending on how they were ordered, it was almost like the words, for lack of a better, or phrases of mushroom neurons, which is just it was weird. It was a lot of math that I didn't really understand, but I mean, just seeing the graphs of it was just crazy. What else you got? So are you going to make a video on this topic or just live streaming? No, I'm probably going to do, like, the plan is to do videos. I'm also going to be doing more Whose Gene Is It Anyway streams soon. I'm not going to be doing it where I like draw an idea from a hat. I want to take the time to really like research the ideas, so that way I can do more like this, where I have all my stuff planned out and it's less rambly. And I you know, I've got my plan. I can just go through step by step, and I'll show you how I do the design and stuff. But I want to have all my material prepped in advance. So I got talking with a lady named Billy Rubin on Twitter, which, by the way, you should go follow me on Twitter. Fair warning, it's a little political. But anyway, we got talking, and she basically asked, is it possible to make a birth control that is genetic? And you know, my initial response was like, that's probably a bad idea, because that's a really good way to like, because birth control is hormonal, and I mean, if you screw that up and you get the dose wrong, like, you're gonna kill someone. But I did have the idea of, all right, so it's not a good idea to actually do to somebody, but could you actually engineer it? And my idea was, could you make a light activated birth control? So basically and then I got thinking about it more, and I'm like, okay, well, if you used a little bit of genetic logic and built like a genetic flip-flop, you could basically make it triggered on a pin code. So like there could be, you could basically do, let's say you like modified your skin like in a specific, like in a specific pattern, so that you had multiple different color sensitive genetic elements that need to be triggered in a specific order to unlock the latch. And only when you unlock the latch does it turn prednisone synthesis off. So that's something that, I don't know. I think it'd be fun to do the design. But like you could make like pin code activated birth control, which I just thought, I don't know. I thought that was cool. I don't know if it's a good plan. Like, I would never suggest anybody actually do this to themselves, but as a thought experiment, I think it's interesting. Also, like, don't do that. Don't try and genetically modify yourself. I mean, period, especially if you don't know what you're doing. But also, don't do it with hormones. Like, that's a really good way to end up dead. Just don't do it. Don't do it. You'll die. Don't do it. I get emails about this all the time of like, could you, do like transition stuff genetically? I'm like, yeah, probably. Don't do it. It's a bad plan. You'll die. Like, it's too hard to control. Like, it's one of those things you really got to get the dose right, and you just don't have, unless you can build the mechanisms in to control it carefully. It's just not a good plan. You have activated pregnancy mode. Yeah, I mean, that's kind of the idea. Like, it's just a thing you could just turn on and off when you want. So that way, you could do the thing, which would be fun. Sounds very unstable. Yeah, I mean, probably not a good plan. We already genetically sterilized crops. I mean, yeah, because people bitch about the genetically modified crops. Like, people complain about the one-shot seeds, but like, it's one of those you can't suck and blow. You can't complain about genetically modified crops propagating and one-shot seeds. Like, they made the one-shot seeds not because they want you to have to buy seeds every year, but because everybody was complaining about what if the GMOs like get into the environment. It's like, okay, well, so we just make the GMOs not replicate. And so then everybody complains about sterile GMOs. It's like, guys, you can't, like, pick a lane. So, that's a thing. Yeah, I mean, somebody like, it's basically the thing of like Oryx and Crake, which I'm not going to do that. I'm not a monster. All right, what else we got? Any other questions about the Neuron project? Thoughts, comments, concerns, quandaries, queries, quintessential quips? I'm out of thing. What else we got? Is it possible to build in control mechanisms for hormones? I know it's a bad idea, but it's theoretically possible. Yeah, no, that's literally what I was just talking about. Yeah, theoretically, still a bad idea. Don't do it. What do you think about atrazine? I have no idea what it is, so I have no strong thoughts. What else we got? Can you make a meat berry again? I mean, can I? Yes. Will I? No. Next time, I'm making kale lungs. You think I'm joking? I'm not. I'm making kale lungs. One of the many reasons I want the incubator working is because I want to be able to get into some mammalian stuff again, and I think kale lungs would just be fucking hilarious. Somebody was like, but why kale lungs? I'm like, well, it's the logical progression between meat berry and vegetable fish. So, any updates on the malarial cancer thing? Yeah, the company screwed up the synthesis. There's a few of the... Okay, so, let's just do a quick little update on some of the 𝐎𝐻's gene stuff that I ordered. The milk and egg constructs didn't work. And they were kind of fundamentally flawed. Like, I made some errors in the DNA design, and also found out that yeast are very difficult to work with. So that project needs to be reworked. Basically, it did technically work. Like, the yeast did produce the milk proteins and egg proteins, but it's at such a low level that I couldn't like brew the cheese omelet that I wanted. So I never made the video because there's nothing to see. Like, I physically cannot produce enough of the protein to actually do the thing. So it's not interesting as a video. And so that's kind of why that didn't go anywhere. I've been meaning to redo it, but like, to do it properly, I'd have to be able to get high level expression either in yeast or in E. coli. And if you do it in E. coli, it comes with all the problems of it's in E. coli. So I haven't really found a good solution to that, which is why I haven't done that one. And the other one that I ordered was the Brainbow yeast, which that also failed in synthesis. That was annoying. And the malarial cancer thing failed in synthesis. So I'm going to be reordering that in a simpler format from a different company. But again, one of the reasons I want the mammalian incubator is so that I can get back on those projects. Like, a lot of these projects were just put on hold because I just didn't have the facility to do them. But I do now, so, Oh, excuse me. I got hiccups now. Man, hiccups are the worst. Anyway. Okay, we just got... Okay, another thing. Any more questions? What else we got? For the Neuron project, are you going to be releasing data recorded, source code, etc. under Commons GPL? Yeah, that's the plan. I'm gonna upload everything. It's all gonna go on GitHub. It's all gonna be open. Again, I see no need to commercialize the thing that I'm making, because ultimately I'm mostly using other people's work. So like, I couldn't patent it even if I wanted. Not that I would, but so yeah, no, it's all going to be open. It'll all be available. What else we got? There's a hair growth process where they inject cells into your scalp. Is it possible to modify these to express fluorescent strands of hair? Okay, so the hair growth thing is called platelet-rich plasma therapy. It actually works surprisingly well, and I'm tinkering with it. We're not getting into that right now. But it's not a way to actually modify the cells. If you wanted to just modify the cells to make like fluorescent hair, that's actually pretty easy. You'd basically want to go in and knock out the melanin production in the follicle cells and simultaneously express some other marker. I mean, you could just do a fluorescent protein or a chromoprotein, and then you could like get blue hair. That'd be pretty easy. But yeah, so yeah, no, that's doable. It's just not, it's not doable in that particular way. But yes, I absolutely, if I were so inclined, could make your hair blue. I'm just not going to, because I mean, don't test on humans. Okay, Yapping Dog asks, would networking them together increase their capabilities? Also, could you train different batches to do different things and link them together? I mean, yeah, theoretically. And I mean, yeah, fuck. I mean, we might try that. Like, that's reasonable. If we can get a bunch of batches growing and we have, like, the limiting factor on that one is going to be the head stage. So either what we'll do is we'll get one head stage with more electrodes and then just, like, split them so that one is connected to one array and one is connected to another. Or just get multiple head stages, but they're expensive, so, like, that's kind of iffy. But yeah, I mean, that's theoretically doable. Mike Ballard just kicked in. Thank you very much. Greatly appreciate it. Update on the magnetic field Hall effect gene mod. There is no update. I put some more thought into it. I might revisit that for a Who's Gene episode, though. Like I say, I want to start doing streams again. Believe me, the only person who's more annoyed that there isn't more content on my channel than you guys is me. So, you know, more streams, and I'm also going to be getting into shorts. I am not like a TikTok person, but I'm going to learn, because that is just the way that the world is going these days. And I think there's so much content that I can make that makes sense in a TikTok format that just doesn't make sense to do a whole video on. So one of the first that I'm going to be doing is I keep a bioluminescent nightlight next to my bed. It's made out of 𝐎𝐵𝖚 fungi, just growing in a jar. I say it's a nightlight. It's not like it provides any significant amount of light, but it's just cool to, like, look over next to my bed every day and be like, oh look, there's a jar of glowing stuff there. That's neat. But I don't think there's enough content there to make, like, a whole video. So that's going to be like a short, maybe either a minute and a half, that kind of thing. But yeah, so I'm planning a whole bunch more short form content that's just, like, quick, easy, snappy. I'm also planning to do a bunch of short form genetic engineering content. So, like, here's bacteria, here's DNA, let's put them together, see what they do, bing, bang, boom, make a bacteria do a thing, like make it green. And or, like, the next one is like, okay, we've got a green bacteria, I'm gonna extract the GFP and turn it into a laser. Like, there's a lot of stuff that I've been working on that I'm just trying to get out and just trying to get the videos done, but they're just getting them right has been the thing that's holding them up for the most part. Like, I'm presently working on an update to the Opal project that I've been working on for frickin' months, and it's just so slow. Like, Opal takes frickin' ages to grow, so it's just a slow project. I physically can't get the video out faster because I just got to wait for Opal to settle, and it takes weeks at a time for every single test. But that video is going to be opalizing bone, so I'm going to be starting some of those trials this week, which I'm pretty excited for. Any chance for a little reveal of what you have planned for the T-Rex bone? Yeah, that's pretty simple. It's basically just proving that there's still viable collagen inside the fossil. There's a whole bunch of papers where they've proven that fossils do actually remain slightly proteiny, and you're able to actually detect viable protein and sequence it even from the fossils. So there's one paper that sequenced the protein sequence of T-Rex collagen and found that it was almost identical to chicken, which is hilarious. But I basically want to replicate some of that. I'm not going to go too hard on it. It's basically, you basically just fix you cut a really thin sample of the fossil from, like, the core, demineralize it, and what's the word? Embed it in paraffin so that nothing moves around, and then you stain it with antibodies to basically prove that there's collagen there, and some, like, collagen specific dyes and stuff. So that's pretty cool. What else you got? Could you make Pinellas produce more glow? Yeah, give it more oxygen, but there's kind of a limit. Beyond that, no, not really. What else we got? What do you think of DNA project using light bug stuff? I assume you mean fireflies. I have a construct that makes bioluminescent glow, like, in my freezer. Like, it works great. I can't actually talk about that project yet. Like I said, I mentioned it earlier in the stream, but I'm not going to talk about that project yet because there's another company involved, and I can't say more. But it'll be ready when it's ready, and it's going to be dope. About the Neuron project, have you thought about directly integrating them into computer architecture like branch prediction? No, that's too hard. The reality is I don't think these arrays are going to be capable of anything even close to that. What else you got? How about growing small pyramid-like structures like the wings on beetles? Also very difficult. Anything that involves growing structures precisely is unbelievably difficult. The closest thing that I would see to that is I've been looking at some circadian clock circuits that basically allow you to periodically produce proteins. So if you're using a motile bacteria or just one that is going to grow and spread on the plate, you can make it produce colors rhythmically. So you can get like a stripe pattern out of a bacteria, which I think would be cool. But in terms of actually growing like structures, that's really hard. There are, however, a bunch of species of bacteria that they've found that do structural color. Like just by the way that the bacteria grows, they automatically line themselves up and are exactly the right size to make an opalescent, like rainbowy sheen to the bacteria. And I mean, I'd love to get my hands on some, but most of them are toxic, so I can't actually grow them because, you know, no growing pathogens. How long can a plate of neurons survive? A surprisingly long time. Like if you keep them fed regularly and maintained, a single dish of neurons can last like more than a month, which is, and I mean, or longer, depending on how good you are at sort of maintaining them and how stable your cell culture conditions are, which I just find wild, because if you tried to do that with fibroblasts, they'd be ruined in a week. What else we got? Bioluminescent construct in plant cells. So a couple of people I know are working on that. And the DNA is available on, and I have the DNA. It's in my freezer. Like I have the DNA for doing bioluminescence in plants in my freezer. The biggest thing with it, though, is you need to be able to do plant cell culture, which I technically have the stuff for. It's just a lot of work, and it's very intensive, and it's not something I'm very good at. So yes, I could theoretically do it, and I might someday, but I have too many other projects going right now to really be able to dig into plant stuff. I want to at some point, but in all likelihood, I'll have moved labs again and hired a whole bunch more people before I can actually get to bioluminescent plants, just because of the nature of how like resource intensive it is. Why no growing pathogens? It's illegal and also stupid, because the most likely person to get infected is you. So, you know, don't grow pathogens. It's a bad idea. Don't do it. Is it possible to run Doom on neurons? I mean, probably, if you try really hard, but it might take you 50 years. Can you make a blinking plant? Actually, yeah. No, that's easy. That's much easier. Like just doing like 𝐃𝐭 in neurons would be pretty feasible. What neurons are you going to use? Human, bug, plant, or what? Well, there's no neurons in plants, so not plants. The plan is to start with rat neurons and then eventually transition to human neurons, just to see if there's a difference more than anything. Like, I'm just curious if you can actually detect a legitimate difference between an array of rat neurons and an array of human neurons, and if one of them is better at solving problems. I don't expect so, but fuck it, I'll try. All right, I'm gonna answer a couple more, and then we're gonna call it for the day because my throat's starting to get sore. What else we got? How does the training process work? I heard the earlier explanation, but I don't understand why the connections are strengthened. So basically, it's a sort of a call and response thing. So neurons will respond by either spiking more or less based on if you're feeding them a high frequency or low frequency signal into one of the electrodes. So if you start feeding in a signal that they don't like, or they respond to, depending on how I say don't like. I mean, they're neurons. It's hard to know what they like or don't like. But if you feed in a signal that causes more spikes to form into a specific, so you feed the signal into a specific spot on the array, and you only turn the signal off when you detect a certain number of spikes in a certain other spot on the array. that's basically how they figure it out. So you just do it over and over and over again, and the neurons pretty quickly figure this out. And you'll want to do this over multiple sessions because, like, as you're stimulating them, it causes them to fire a lot more, and so it uses up the food in the media and screws with the balance of the media more. So you can only do a couple, like a short, brief experiment at a time. But if you regularly do the experiment, so every couple of days, you do the experiment, you feed in the same signal into the same spot, and you only turn it off when the spikes show up in the right spot, they will adjust. Like, they will readjust themselves so that they do the thing that you want. It's crude. It's very crude. But, I mean, that's sort of what you get when you're working with something like this. All right, what else we got? Is the blinking plant an oscillator, or running some kind of cycle? Like, would like to see DNA programming? Blinking plant would either be based on a circadian rhythm thing, so it's really only turning on and off versus like when. Okay, dude, just chill. So it would only be turning on or off when you like shine a light on it for a certain amount of time, or you build in an oscillator that just oscillates much more quickly. But the problem with this is, how do you control it so that the oscillations are slow enough that you could actually like see something? The alternative is you do something like the 𝐔𝐸𝐿, which let me just go back to that for a second. Like, you could do something like this. Like, this is really easy because it's literally one protein, and just expressing one protein in a plant is like hilariously easy. And especially because you could just do this on a constitutive promoter, something like cauliflower mosaic virus promoter, which is nice and easy. It's quick. And then you could just have a UV light shining on the plant, and you would see it pulse like this. I don't have the videos available right now, but there are some videos like where if you cut a leaf off, you'll see a flow of calcium. And that's the way that the plant recognizes that there's damage. And so there was another video from the same group that did this, where they put a caterpillar on the plant, and you can see that every time the caterpillar takes a bite, there's like a flash of light from the calcium being released. So that's pretty cool. Okay, I'm going to take 2 more questions, and then we're calling it for the day because my throat's killing me. Could you do a repeating signal like an SOS in Morse code? I mean, yeah, theoretically. One of the things that I really want to explore on a Whose Gene series is genetic logic. And because if you could build logic gates genetically, then you can actually do things like simple counters and stuff where, you need the signals to come in a certain order for it to like trigger a series of flip-flops. Because if you can make one flip-flop, great. Now you just make a series of them. And of course, this is very tricky with genetics because every single flip-flop requires a different trigger, which is why it's much harder to do this genetically than it is to do it electronically. But if you could get a series of flip-flops, then you could just have it like flip, 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 and have like a different color turn on when each flip-flop is on, or just like turn on expression or something. But you could have it so that the output of one feeds into the next to trigger the next flip-flop so that it just ticks circularly. It'd be hard, but it would be doable. All right, last one. Let's pick a good one. Could edible glowing fruit be engineered? Yeah, actually, this is pretty easy. So, again, I can't pull it up because I'm sticking to only presentation this time because I don't want to open up another Chrome window. But there's a company called light-bio.com. Like, if you go to light-bio.com, there's a company that's engineered a suite of bioluminescent plants now that are going to be available for sale soon, hopefully. They've got petunias, a tobacco derivative, and I think an English ivy and a few others. And so there's no reason that you couldn't do this in like apples. And theoretically it should be food safe, but I mean you'd have to actually do like first animal testing and then eventually human testing before the FDA would ever allow you to like sell that as food. So yeah, it's very doable, but not Not necessarily something you want to just like jump to. But all right, I'm gonna call it there. Thank you everybody who donated and who came and hung out. This has been a lot of fun. I hope you guys are as excited for this project as I am, and I'm really excited to get going on it. And there's going to be more videos, more streams, more shorts, all kinds of stuff. So as always, be sure to subscribe if you haven't already, and I'll see you next time. So thanks again, everybody, for stopping by. Have a lovely rest of the evening, and I'm out. Bye.